Hello, everybody. How are we doing? Good to be here. We are talking today about the reproductive system. It's a good one. It's a real banger of a system. And I wanted to talk kind of at the beginning about a couple things. We're going to talk about fertilization, but I wanted to talk about kind of a hot thing, this era that we live in with, our ability to kind of use biotechnology and in vitro fertilization puts us in a kind of interesting time frame. I want to talk a little bit about designer babies just to kind of start this off. And like something you can already do now is with pretty good accuracy is gender selection in a pretty simple, straightforward way. You can do it with 100% accuracy in another method. So, we could pick the gender of our kids. I mean, I guess you could really do this in a couple ways, but one of them would be pretty unethical. I'm a little concerned about all this because if we're designing babies, isn't this a form of eugenics? The thing Hitler did, where we're trying to select for the ultimate human in some way. It's a pretty interesting kind of topic. I'll leave you that up to you to decide. I've got a couple of ideas for the discussion post, but we'll see if we do it on that. Anyway, gender selection. So one way that we could do this is if you look here, we have a female. So we have 23 chromosomes as human beings. 1, 2, 3, 4, 5, 6, 22. These are our somatic chromosomes or our body chromosomes. And then we have our sex chromosome. And this is a female, has 2 X's. Whereas over here, boo, 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 XY, we got a male human here. And this is called a karyotype. So you take a cell and you crack it open. Crack open the nucleus, which is its karyote, right? Eukaryote. Karyotype. And you can see what we're working with. Well, one way that we can kind of do gender selection or increase the chance of having a baby is we can look at the sperm. Because the sperm, yo daddy determined your sex, whether or not you were male or female. Because the dad, as you can see over here, can either donate an X or a Y. Your mama could only donate an X or an X. That doesn't determine gender, right? You have to have, if the dad gives a Y, boom, then the kid's a male. The dad gives an X, boom, the kid's a female. So your daddy determined your sex. So they can take the sperm and they can add a little fluorescent dye. So gender selection. Your daddy determined your sex. The sperm looks something like this. Very simply. And if it's got 2 X's in there, plus everything else, you can see these X's are actually a little longer. I'll do it something like this. Right? And we add a little fluorescent dye, it's going to be a little bit brighter than if we have one that is X, Y. Because it's smaller. So this is going to have a brighter fluorescent signal than this. This will be a dimmer signal. So female, so we determine your daddy's sex, we add a DNA dye to sperm. Female sperm will be brighter because the X, and really they would only have one of these, like that. female sperm will be brighter because the X chromosome is larger. And male sperm will be dimmer because the Y chromosome is smaller. So we can sort the sperm based on brightness, and this gives you a pretty good chance of then you can take the male sperm and fertilize the egg with the male sperm, and it's more likely that you'll have a male child that way. And then you can implant that baby into the mother via implantation, like in a fertility clinic. This would all be kind of done at a fertility clinic. The other thing we could do, okay, is we could take our sperm. We could take our sperm, and we could take our egg, and I'm going to go into what this is, and we can mix them together. In a second here, we can mix these together. And what happens is we get an embryo then. Okay? And it starts to divide. And so now these cells are dividing. And let's say it's got like, I don't know, that many cells now. Okay, what we could do is we could pluck one of these cells off. Bloop. Let's take this one. Pluck a cell off. And we can do a karyotype on it, or we can sequence the DNA on it. Okay, so let's pluck that cell off. And the embryo will develop fine, actually. If it's early enough, if it's at the 8-cell stage or less, I believe, kind of depends. You can take a cell off of an embryo and it'll still develop fine. And we can sequence this DNA. We can see what genes are contained in here. We can do karyotype to, for example, look at whether or not they have Down syndrome. Do karyotype to look at the chromosomes, or sequence the DNA to see what genes it has. And if we like the genes that it has, we can keep it, and it will continue to develop. Okay? This will continue. If we like the genes it has, we can implant this and allow it to develop into a human. Okay, pretty crazy stuff, honestly. Okay, I don't feel that great about it. It concerns me. Because now we got this human walking around with, let's say, blue eyes. Okay, and maybe we also selected genes for them to be really tall with weird feet and long arms. All right, and a big butt. Okay. It's really kind of strange. It's a weird time that we live in because we could do stuff like that. Yeah, you could literally do this. You can select the eye color of your baby now through a fertility clinic. Another thing that happens that's interesting. So, I don't know if anybody lets you select a big butt baby, but right now you can select eye color, which is actually a couple different genes control that. Select the eye color of your child at fertility clinics. crazy stuff. I actually learned about that from a student and then looked into it. It's crazy. I didn't know we were that insane yet as human beings. Okay, the other thing that people do is you'll have a child. Draw this real quick. You'll have a child with a genetic disorder. And they could treat it with stem cells, let's say. I'm trying to think of, my mind is not really working right now. You have a child with a genetic disorder or something, something pretty uncommon, but we do have a method for kind of alleviating it if we have somebody with similar cells. For example, a second child can be made to harvest stem cells from to save the original child. Okay, so then some parents will have a second child to save the first. I don't really know if there's a name for this, but like a donor child, essentially. Because like when the baby's developing, like we have an umbilical cord and harvest umbilical stem cells to treat the first child's disorder. I don't have the time to go into like the details of that. Gene therapy. That was the kind of term. It's very similar to gene therapy to treat first child's genetic disorder. Like let's say, for example, they have like some kind of like rare blood cell disorder or something like that, and we need blood stem cells. We could select a child that would be a match via in vitro fertilization for the first child that wouldn't have the disorder, right? We could do this same kind of method here, right? Where we pluck up the cell, make sure they're okay, the second child, and then we could harvest their bone marrow cells and put them in this first child, for example, something like that. You know, I'm sure there's a lot of other diseases. That's just kind of one example I thought of. Okay, so we live in a kind of crazy time where we're allowing parents to be Hitler. Pretty weird. Okay, I want to talk real quick also in this one about this process. This process here is called fertilization. Fertilization. When a sperm meets an egg, it fertilizes the egg. Okay, so what does that actually mean? Well, sperm are actually really tiny, so we're going to draw a big old massive egg here. Okay, you might not want to draw it as to scale as I'm doing it. This is an egg, and it's haploid. We've not talked about those terms, but I will when we talk about mitosis, meiosis. So every chromosome, you have 2 copies of it. You see chromosome 1, we have 2 copies of it. 2, we have 2 copies. 3, we have 2 copies, right? Our sex chromosomes, we have 2 copies. When we make a sperm and an egg, we throw away one of the copies so that when the sperm and the egg come together, we now have 2 copies of everything again. Okay, so these would be diploid, di meaning 2. Let me put this here. Diploid is the normal chromosome number for our body cells. Okay, diploid. So if you were to take your skin cells, hair, eye, butt, intestine, all of that, they're diploid, meaning two. And then ploidy is chromosome copy, chromosome number. 2 chromosome number. Okay, 2 chromosomes of every one. So we have 2 versions of 1, 2 versions of 17. Okay, haploid. Haploid's like half. Haploid is the number of chromosomes in our gametes, sperm and egg. That's what those are, right? Because we want them to be, when they come fused together, we want them to be diploid. So this means 1 or half, and this means chromosome number, right? Okay, those are some terms you need to know. All right, so this egg is haploid. It has 1 of each chromosome, and it's waiting for daddy to come to town. All right, and our sperm anatomy here, let's do a little sperm anatomy. Sperm are really cool cells. They look kind of like little triangular ghosts. Maybe drew that a little thick, but that's okay. Something like that, okay? And then here, we've got a boatload, a very specialized organ called mitochondria. Mitochondria. Got mitochondria there. And then here is where all the genetic material would be. And we're just going to draw a couple of them. Let's draw 2. We'll draw a green one, and we'll draw a blue one. This would be our DNA. This would be our DNA. And this is, again, a haploid sperm. Haploid sperm. All right, and some anatomy terms. This is the tail. It's a flagella. Flagella, and it whips around to allow the sperm to swim. Swim. Okay. Mitochondria obviously fuel the ATP. These make ATP. These make ATP to whip the flagella around, to allow the sperm to swim. They got a long way to go, man, all the way up through the uterus, up into the fallopian tube. It's pretty crazy, okay? All right. That's the tail. This is the mid piece, where all the mitochondria are, mid piece. And then this is the head of the sperm. And the very tippy tip is called the acrosome. Acrosome. And this has a lot of, like signaling and enzymatic activity. It binds to the egg, signaling and enzymes to fuse with the egg. Okay, so it's kind of decorated with proteins, and it releases a lot of, put it up here, enzymes to kind of chew through the outside of the egg. Okay. All right, another term for this, like I said, is this is a gamete. This is a male gamete. All right, these are made, we'll talk about where they're made. Okay. And now over here we've got our female haploid egg. And it's got, the sperm has a short chromosome and a long chromosome. So we're going to draw a short chromosome and a long chromosome over here as well. All right, so we'll do the short one and we'll do it in red. Short in red, and long we'll do it purple. Okay, so it's also a haploid egg. Got two of each, or it's got one of each chromosome, short and long. Okay. All right, what happens? Well, what happens is the sperm, I'm just going to kind of copy this to cheat a little bit and erase some of this. It's going to find, when it finds the egg, binds to it, and if everything works out good for the sperm and the egg, what happens is this, there's like a jelly coat around the egg and all that. A lot of sperm will find it. You got to be the lucky one to be able to get inside. It'll chew through the egg and it'll fuse with it like this. Okay? And it will inject its chromosomes, like so. And they will come here into the nucleus of the egg, and now we've got that one and this one. Boom. So now we've got two short, two long. This egg has been fertilized. Boom. So now actually, let me do it like this. Haploid egg. Put that down there. Get rid of, it was that and that. This becomes a diploid embryo. And that happens after fertilization. After fertilization. Okay, and then this is going to start to divide. Rounds of cell division will then occur. This will start to divide. It will develop into a human. It's pretty crazy. All right. And then this is, actually this embryo is also called, another term for it is diploid embryo or a zygote. It's now a zygote. Boop, 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 boom. This was a gamete, the sperm is a gamete. And this is also a gamete. Okay, so that's basically how fertilization works, okay? That's what's going on there. Very simply, straightforward, is we've taken our DNA, we've split it in half to make our gametes. We fuse them together to make our zygote, to make our embryo. We go from an egg, that's the difference between an egg and an embryo. An embryo has been fertilized. Okay, and that's going to signal to this cell to go through all these divisions. And let's see if I can actually pull up a little video. Yeah, we'll pull up this little video here of a salamander kind of going through development after fertilization. Okay, so that's the first cell division right there. So this was fertilized. Boom, boom, boom. And then boom, first to cell division. And it's going to go through another one. So now it's a 4-cell embryo. Now it's 8-cell, 16, 32, 64, 128, 256, 512, and now, so you notice these were all synchronous, actually. They divide about every 30 minutes. I studied frogs, so I know how this works. Every 30 minutes they divide synchronously. And we'll talk about this process of cell division and how this works. And then after a while, after they get to like 512 or 256, they start dividing asynchronously. The cells start to divide kind of randomly all throughout. This is very similar to how humans develop as well. Then this part here is called gastrulation. The cells start to migrate and move in and fold in on the embryo like a popcorn. So those cells are crawling inside to form the different tissue layers, like your digestive system. And this right here is going to form your spinal cord. It's called nerulation. So it's doing this again. It's folding in on itself. So this is going to be the head of the embryo, and this is going to be the spine. This is forming the brain up here, and this is forming the spinal cord. Right here is going to be the head and the eye. This is why amphibians, so you see these guys, these are cells that are crawling now from this thing called the notochord in the back where the spine's going to form. They crawl around and they pull on the stiffness of the matrix based on how much they like to pull. They'll settle, and they'll become like, if they like to pull a lot, they'll settle on stiff things like collagen, and they'll become a bone cell, for example. Or if they like to crawl around onto soft things, they'll become a brain cell. They'll hang out with just like an elastic fiber that's really soft, and then they'll become a brain cell. And then this embryo, you can see, is kind of breaking out of its little membrane. It's continuing to develop. You can really see its head. You can see some pigmented cells here on its back. These are little pigmented cells. We got some blood cells right there. You see those pigmented cells? They're like little octopuses. They're little neurons, but they're just pigmented cells. They hold pigment, just like melanin in our skin. You can see its heart beating. Those are its gills right here. Those are allowing it to breathe. So you see those blood cells going through the gills. Its eye is developing. That all started off as a sperm and an egg, and then a single cell. And through tiny little cellular forces of cells dividing and crawling and pushing on each other, we've developed into this whole organism. I have like extremely bad back pain right now. It's really messing with me. But that was beautiful. Alright, I'll see you guys in the next one. Later.